For the present video is cleaning validation guideline in the pharmaceutical industry. This video is for the guidelines related to cleaning validation and provided by different regulatory authorities and different regulatory bodies. This is a short video for cleaning validation guidelines and what is the main focus of these guidelines. First of all, we will see what is cleaning validation. So cleaning validation is a type of validation, or you can say it is a validation process performed in the pharmaceutical industries which ensure that the manufacturing equipment is clean to remove the residues of previous products, contaminants or impurities to such levels that these are safe and do not affect the product quality, safety and efficacy. In pharmaceutical industries, many of the different products are manufactured in the same area and using the same equipments. So their proper cleaning before starting the next batch or after completion of the batch manufacturing is very much essential, and this is ensured by cleaning validation process. For example, you consider that you are using a granulator in the area for making some formulation. Let's say you are working onto the batch of diclofenac. You completed the batch of diclofenac tablet. You have cleaned the equipment and area, and now you are going to use the same equipment for making the telmisartan batch. So diclofenac batch material, excipients or contaminants should not come into the telmisartan batch, and that is ensured by the cleaning validation. Many of the times potent molecules are prepared, potent drug substance formulations are manufactured along with some non-potent type of APIs. So that time it becomes the critical thing to manage those contaminants, and that's why cleaning validation is important. Cleaning validation aims to verify that the cleaning procedures are sufficient and cleaning procedures are ensuring the effective removal of residues of the active pharmaceutical ingredients, excipients, and also the cleaning agents and any potential microbial contaminants from the manufacturing equipment, so that it is not going into the next product. Ensuring that the products are safe. Importance of cleaning validation. So it ensures GMP compliance in the regulated manufacturing. It minimizes the cross contamination risk and maintains the product integrity. Transition to risk-based, science-based validation models and HBEL, that is health-based exposure limits. So these are some of the important points for cleaning validation. MACO value is related to cleaning validation, that is maximum allowable carryover limit, MACO value. Nowadays, the HBEL based calculations are being done. Recently, the cleaning validation trends are being shifted from traditional V model validation to risk and science-based models. So adoption of HBELs for setting the limits. MACO values are also used, but now HBEL is used for setting the limits. Updated guidelines by major regulatory authorities are there from last 3-4 years, 2018 to 19. So as per FDA, CFR 21 CFR 211.67 the guideline is revised. And the key requirements are like equipment must be sanitized or sterilized to prevent the contamination. There should not be any residue, so no acceptable residue levels aims for complete removal, and TOC related information is there. So total organic carbon, that is TOC, accepted for residue monitoring. Continuous process verification is essential for routine checks. So as per FDA, you can find different guidelines which are having the key requirements of TOC, residual limits, and continuous process verification. Important links like you can search and find like FDA cleaning validation guidelines and question and answers on current GMP practices. So here you can get all the information. Coming to European Medicines Agency EMA, guidelines are effective from June 15 and mandatory HBEL are all for the all products. So focus areas are established HBEL to safe residue limits. Distinction between acceptable limit and alert limit is also given there. Analytical testing required during product changeover, and LD50 data cannot determine the HB. Useful resources you can have like EMA guideline on HBL and question and answers on guideline implementation. WHO has given some guidelines similar to the FDA guidelines, and here they consider worst case approach for selecting representative APIs, continuous process verification with analytical and visual examination. So analytical verification is required. Focus is risk-based methods for cleaning validation. This is regarding WHO. Now PICS is also there, that is Pharmaceutical Inspection Cooperation Scheme, and they have guideline EI 0461 which emphasizes on HBEL in the shared facilities. So shared facilities means the manufacturing site where many of the products are being manufactured. So these are the shared facilities. Key resources are the PICS guidelines on cross-contamination in shared facilities and guideline for quality risk management in cleaning validation. EGA has given some guidelines, adoption of PICS guideline for cleaning validation, which have key points like transition to risk-based SOPs, continuous process monitoring, and alignment with the global standards. Health Canada has guideline which emphasize on actual versus worst case risk, what is involved in the cross contamination, dedicated equipment for specific products in multi-product facilities. So these guidelines also give idea and information on the dedicated equipments for specific products. Revalidation triggers, that means when the revalidation is triggered, which is based on the changes in the cleaning procedure and the raw materials. So this is regarding the Health Canada. Now PDA also given some information and details. Parental Drug Association, that is PDA, it has technical reports on cleaning validation for active ingredients, cleaning validation for biotechnology formulations of products, and it recommends that review both reports for detailed validation SOPs and templates. International Society for Pharmaceutical Engineering risk map is there, which aligns with EMA guidelines on HBELS. Cleaning validation life cycle guideline or guide, which include application methods and controls. APIC guideline is there. Active Pharmaceutical Ingredients Committee, that is APIC. This focus on cleaning in API plants, and it provide you the information on challenges, consistency issues, complex terminology, and residue limit calculations. American Society for Testing and Materials, ASTM guidelines are also there. ASTM E3106. It is a standard guide for science-based and risk-based cleaning process development, and it also provides you future standards for visual inspection, cleaning, validation, limit calculation. So many guidelines are there. You can search these guidelines. I have provided you all the important material for searching the guidelines, and also I have provided the guideline, and you can search and read those guidelines in the detail. I will be making upcoming videos on the detailed guidelines on HBEL, on MACO, how to calculate, what is risk-based cleaning method, what is science-based cleaning method development, what is MACO, what is worst case. So all these videos I will be making. So stay tuned with this channel. So in conclusion, we can say cleaning validation is evolving with strong emphasis on risk and science-based methods. Compliance with global guidelines and periodic re-evaluation is very crucial. So we should stay updated with each region's regulatory expectation to ensure GMP compliance. Because nowadays regulatory guidelines are coming on cleaning validation many times. Many of the times it is noted that these guidelines provide more clear information, detailed information for the cleaning validation approach. Then we should also be very updated with the guidelines provided by major authorities like US FDA, EMA, TGA, and Visa. So these guidelines can be implemented, and also consider the product potency, product therapeutic category, and whether it is whether manufacturing of that product is related to the health hazard or not. So I hope this video will help you to understand these things, and in upcoming videos we will talk more on the training validation. Thank you for watching the video, and I request you to share this video with your friends and colleagues. Thank you.